right, so this will be the first lecture, but I'm going to give you a little bit of repeating of my introductory stuff, just so I've heard it at least more than once. So as I told you yesterday, my name is Mike Hannigan. I'm a graduate of Holton High School, St. Joseph's College. I have a PhD from University of Connecticut Health Center in Farmington. My research was in immunology, but I have been teaching anatomy and physiology for Northern Maine Community College for 18 years, and I've been working at Holton High School for 20 plus years. So to break down your work, your text again is Anatomy and Physiology, the Unity of Form and Function by Saladin. It's available as an ebook, and it comes with your Connect Code purchase. Homework is going to count 35% of your grade, should be completed weekly. Put deadlines of 2 weeks, but again, I do not penalize for late work. Labs are 20% of your grade. They will be posted each week. This week it should be going up about Thursday. Again, it should be completed weekly, but there is no penalty for late fees. Tests will count for 30% of your grade. I think we have 4 of them scheduled throughout the semester, but they do need to be completed within the posted time frame. No late acceptance of testing. And participation, attendance, if you're in person, would be 15% of your grades. Again, if you want to contact me for either Bio 20121 or 20160, best way to do it is my Northern Maine email contact, mhannigan@mainecc.edu. Pay attention to Brightspace and reach out when you have questions. So today we're going to start the introduction to anatomy and physiology, and we're going to talk a lot of basic biology here. Building the groundwork for the science behind what we'll be dealing with for the next 2 semesters. So anatomy is the study of structures in the body. Physiology is the study of the function of those structures in the body. They are complementary to each other. The shape of something, the anatomy of something is going to determine what it can do, so it's going to, in a great deal, determine its physiology. I always like to say that spoons are great for eating soup. They're not so good for eating things like spaghetti, and that has to do with their structure and the function you want to carry out. So physiology provides meaning to anatomy, and anatomy is what makes the physiology possible. There are a number of ways that anatomists have looked at the human body through the years. Some are more recent than others. We've always had inspection just to look at an appearance. If you hear the term palpation, that is to feel the structure with your hands. Some of your lab work, you'll be asked to palpate muscles as they move. Auscultation is listening to sounds produced by the body. The stethoscope is a very important tool for physiologists, physicians, as well as other listening devices. When you add a tap on the body, you might be feeling for resistance or listening to an emitted sound that's different if there's a pathology in place. We call that percussion, just like a drummer in a band. Dissection, of course, is cutting into, separating the tissues to reveal the tissue relationships. Often done with a cadaver. A cadaver is a dead human body, and that has not been done for as long as many of us probably think. It was a relatively new discovery back when medicine first became a science. We'll get into that in a minute. Comparative anatomy is to use all of these things, particularly dissection, to learn about to compare the anatomy of one organism to the anatomy of another, and that can help us with a lot of information. A lot of things can be done in other organisms that we wouldn't want to and shouldn't do in humans. So exploratory surgery would be to open the living body to see what's wrong. A lot of times that can be substituted for medical imaging so that we can get inside the body without actually cutting into the body. And one of the sciences that deals with that is radiology, but it's not just x-rays anymore. So anatomy is the study of the form. There are numerous ways we could approach anatomy. There are many subdisciplines within anatomy, starting with gross anatomy. Those are the things that we can see just by looking with our naked eye. If we get down to the level of tissues or cells, we have to use histology. And histology is the microscopic examination of anatomy. When we are looking for signs of disease, that becomes histopathology, and I already used the term pathology. Pathology indicates a disease process going on in a human or in another organism. And if we want to look at individual cells, which that's the way medicine is going, then we are into the study of cytology, a single cell, the ultrastructure within a single cell, the biochemistry that's going on within a cell. And a lot of times that cannot use a simple light microscope. Many of those structures are only visible under an electron microscope or other newer microscopes. Physiology is the study of function. Physiology uses methods of experimental science, which we're going to get into a little bit in a moment. There are subdisciplines of physiology, including three big ones would be neurophysiology, endocrinology, pathophysiology, so physiology of nervous system, hormones, mechanisms of disease. And comparative physiology is a very important science as well. We can learn from different species and compare them to our human physiology. So a lot of our understanding of human physiology and the development of new drugs and medical procedures takes place in this big realm of comparative physiology. Does it work in vitro? Does it work in mice? Then will it work in primates? And then we try it on people if it passes many of those tests. Okay, now we get a little bit of history, and this course is not about history. This is to give you a better understanding of the background behind what you're going to be dealing with for the next couple of semesters. So if you're a history buff, there's tons you can find on all of this. But like I said, this is not a history course. I'm just going to give you some stuff that I think is important for you to have an understanding of the background. So we'll start with Hippocrates. You've probably heard of the Hippocratic Oath. Well, Hippocrates was a Greek physician called the father of medicine. He established a code of ethics, which we refer to as the Hippocratic Oath, and he urged physicians to seek natural causes of diseases rather than attributing them to the acts of gods and demons. And this is a huge philosophical shift in many people's beliefs at the time. Aristotle, however, believed diseases had supernatural or physical causes. If they were caused by supernatural, he would call them theologi. If they were caused by natural causes, he would call them physiologi. And these terms gave rise to the term physician and physiology. He also believed that complex structures were built from simpler parts, which is really an early delving into anatomy. We go way ahead to the 1500s. We have Andreas Vesalius, when the Catholic Church relaxed some restrictions on dissecting of cadavers. He began to perform his own dissections rather than having the barber which was also known as a surgeon, do the dissection. And he published an atlas of anatomy, which I am not going to try to read the Latin name, but on the structure of the human body in 1543. William Harvey was a very important physiologist. He contributed to the birth of experimental physiology. His book, in English, On the Motion of the Heart in 1628, is the first time that people realized blood was a circulation, that it left the heart and came back to the heart. And he was not alone in this. There were others who contributed as well. The real beginning of the cell theory, once the microscope was invented, and this is many, many years, this is 1800s, so 300 years later, was Schleiden and Schwann. One of them worked with plants, one of them worked with animals, they worked with other people, and they came to this staggering conclusion that all living organisms were made of cells. And that became the first tenet of the cell theory, considered to be the most important breakthrough in biomedical history. Not only are organisms built from cells, all the functions of the body are effects of those cells, together or alone. It was huge change. That led to revolutions, beginning of the germ theory of disease, that diseases were actually caused by invisible organisms that could not be seen without a microscope, and some of them can't be seen with a microscope, things like viruses. These were what was causing disease, and that eventually led to ways to specifically treat diseases. The mechanisms of heredity were discovered, first by Mendel and then rediscovered and figured out. That led to the structure of the DNA. Advances in medical imaging have come continuously and are still coming. Disease imaging and treatment has changed greatly. Mapping of the human genome promises to change our view of medicine even more in the very near future. So how did this all come about? The most important thing for you to know from here is this experimental physiology is using a scientific method, which is nothing more than a process of performing a science which includes careful observation, logical thinking, proper analysis of observations and conclusions. Scientific methods are a way to show if something is true or false. There are a couple of different ways you can do it. One would be the inductive method, and that would be making numerous observations until you become confident in drawing a generalization and a prediction. Most of the anatomy information would have been obtained in this method. So you observe it over and over again, then you're going to generalize it. And in fact, when we get into anatomy, you'll see that we are generalizing a lot of things all year long. So if we want proof, we need reliable observations that are repeatedly confirmed, not falsified by any credible observations. Once you show it's not true, then it's no longer true. So again, as we are probably all aware, especially if you were around during the pandemic, that in science, truth is tentative. Proof beyond a reasonable doubt. If something comes up that disproves what we believe, that we have to move on and change what we believe. A lot of physiological knowledge is gained by the deductive method. In this process, an investigator formulates a hypothesis, and that hypothesis is an answer to a research question. It's an educated guess based on what you know. So a good hypothesis is consistent with what's known, and it must be testable. And part of that testability must be that it is falsifiable. If you can never prove that something is not false, then you can't prove it's always true. So if we claim something is scientifically true, we must be able to specify what evidence it would take to prove it wrong. There are a lot of experiments that go into this. This is an experimental science, and things that are going to contribute to good experimental design include sample size, the number of subjects in a study. The more subjects, typically, the more likely you are to accept that conclusion. The controls groups in a perfect world are exactly identical to the treatment group, except for that one thing, the treatment in this case. But that's very difficult, especially in humans, so you want to make sure that it's as close to that as you can get. We also have to deal with psychosomatic effects. The placebo effect is a real effect. We don't completely understand that. And one way to do that, to avoid that, is to use the double-blind method, where neither the person administering the drug nor the person receiving the drug knows whether they are receiving the drug or the control substance. And statistical testing will come into play. Things like t-test to give you an idea, based on all of these things, how confident you are that your answer is correct. Science undergoes peer review, which is the critical evaluation by other experts in the field. That's done prior to either funding or publication. It's done by using verification and repeatability of results. It helps to ensure honesty, objectivity, and quality in science. And it is a vital part of being able to trust what you see in scientific publications. So in science, we have facts, laws, and theories, and we tend in our general rule to interchange some of these words, but in science they have a very specific meaning. A scientific fact is information that can be independently verified. A law of nature is the results from inductive reasoning and repeated observations, written as a verbal statement or mathematical formula, and it is considered always true. A law exists until somebody can prove it doesn't. If anybody ever throws something up and it doesn't come down, the law of gravity would have to be rewritten. Most of what we deal with in science are theories, and theories are an explanatory statement. It's a hypothesis that's been confirmed over and over again by many different people in many different ways, but for some reason it cannot be stated that it is always true. And most of the time when we deal with theories, we deal with, in the general public, a theory is just a good guess. Well, in science, a theory is something that's probably a law, but we haven't found ways to experimentally prove that it's always a law. Theories allow for predictions in science, and if any of those predictions ever don't come true, we have to rewrite the theory. We'll touch on evolution, natural selection, and adaptation because we are the products of evolution, selection, and adaptation. To understand biology requires you to understand the theory of evolution. Evolution is a change in the genetic composition of populations of organisms, so developing bacterial resistance to antibiotics by some bacteria is an example of an evolution. The driving force behind this is mutations and natural selection. Natural selection means that there are selection pressures out there that promote the reproductive success of some individuals more than others. So predators, if something comes along genetically that gives you an advantage against predators, you're more likely to be able to reproduce than those who don't have it. And when you reproduce and pass on those inherited features that you have randomly generated, we can get adaptations. These are changes in our anatomy or physiology that have developed or have expanded in response to the pressures that enable that organism to succeed. So if your coat color hides better in your environment than other members of your species, the predators are more likely to get the other members of the species, and your coat color gives you an advantage, and that advantage is always to be able to reproduce more successfully. That theory of natural selection, with the explanation put forth by Charles Darwin, an influential biologist, presented the origin of species by means of natural selection in 1859. Another book, The Descent of Man, in 1871. All right, now let's get into human structures. We're going to go through and list some human structures, most complex to simplest, discuss both reductionism and holistic and clinical significance of variation. So a human organism is based on successive levels of hierarchy. We have an organism being the entire organism, the entire individual. That individual is composed of organ systems. Those organ systems are composed of organs. The organs are composed of tissues. The tissues composed of cells. The cells are formed from molecules that form organelles, and those molecules are formed by the joining of atoms. So an organism, a single, complete individual. Organ system, a group of organs that do a unique function. We have a circulatory system, a respiratory system. In fact, in Anatomy and Physiology this year, we'll look at 11 different systems. Those systems are made of individual organs, 2 or more tissue types that work together and carry out a function. An organ has a defined anatomical boundaries, and you can have organs within organs. Tissues are groups of similar cells that produce products that form a discrete region of an organ and perform a specific function. We're going to look at the 4 different types of tissues in a very near future. Cell is the smallest units that carry out all the basic functions of life. And within those cells are organelles, things like mitochondria, ribosomes. Those organelles are made of molecules, many of them, proteins, fats, nucleic acids, carbohydrates. Those are called macromolecules. And those are made up of atoms, and those atoms are the smallest particle with a unique chemical identity. There are 2 complementary approaches to studying almost anything, but in this case we'll talk about human life. We can use a reductionism or a holism. Reductionism means that we look at the large complex systems, but do so by understanding their simpler components. This is really how we're going to approach anatomy this year. We're going to go 11 different body systems, one at a time, and break them down into simpler parts to see how they work. And each time we talk about an organ system, we'll probably tell you it's the most important system in the body, even though it's not any more important than any of the other 10. You can lose the picture. This is where you're in danger of losing the sight of the forest for the individual trees. A holistic approach or emergent properties as we go to we could look at the whole thing, and really that doesn't work for learning about anatomy and physiology at this level. It does have important implications, and it is an important way to look at things, but reductionism is the method we are going to be using. Now, there are variations in humans, even though we'll tell you in your text and as you're going through that every human has 206 bones. No 2 humans are exactly alike. Even identical twins have differences. So when we talk about the anatomy, we show the most common organization of structures. There are some individuals who don't have certain muscles. There are some individuals that have a different number of vertebrae than the average. Some individuals have an atypical number of organs. Kidneys, for example, are one. Some individuals can show things like situs inversus, where your left-right reversal of an organ, left what's normally on the left, or what's on average is on the left, ends up being on the right. Here are a few examples of different kidneys that are not common, but do occur, and branching of the aorta is another example where we have variations. So life is a collection of properties that distinguish living from non-living things. Characteristics of life, as we will describe them here, include organization. They have higher levels of organization than non-living things. They have a cellular composition. They have metabolism. They are responsive, which means they are excitable, and they are responsive to things that happen in the environment, which we would call stimuli. Organisms, living things have movement, and even if they do not have locomotion within individual cells, needs to be the movement of materials. Homeostasis is a ability to maintain a relatively stable internal condition. Living things develop, they change in form and function over time. That change is often called differentiation. Differentiation is the transformation of unspecialized cells into cells with a committed task. Organisms display growth, they increase in size, and this is a result of chemical changes. Organisms reproduce, they make copies of themselves and pass those genes to offspring, and that contributes to evolution. Organisms have adaptations that change over time based on selection pressures. Physiology is even more variable than anatomy. When you hear normal values, you will always hear them within a range, and I don't even like to use the term normal. I would consider them the average. There are variations that are due to sex, age, diet, weight, physical activity, genetic, and environments. So a reference man would be typically the average 22-year-old, 154-pound light physical activity who consumes 2,800 kilocalories a day. Woman, you would use the same, except you would change the weight to 128 and 2,000 calories a day. That becomes important because, again, it's just a generalization, and if you are a physician or in the healthcare industry and you don't take into account your patient versus the average, you can end up with a lot of over medication or under medication. And one of the biggest things that's really coming around now, beginning to be corrected, is that much of our early research was done on cohorts of men, and there is variation between the sexes that needs to be examined and accounted for. So here we come to homeostasis, a closer look at that ability to detect change and activate mechanisms that oppose it, thereby maintaining a relatively stable internal condition. One of the first to note this was a gentleman by the name of Claude Bernard. He noted that there were fairly constant conditions despite external conditions changing, temperature, for example. Walter Cannon then coined the term homeostasis, which would be the ability to keep those internal conditions the same. Now, there are two different mechanisms that can be used to maintain homeostasis: a negative feedback mechanism and a positive feedback mechanism. What you really need to get out of your mind is the connotation of the word negative. Negative feedback is a good thing. Negative feedback allows you to fine-tune the set point. So internal conditions are going to fluctuate within a limited range. So what we really have is a dynamic equilibrium. It goes a little above and a little below the set point. And negative feedback is the perfect way to do that. This is a mechanism that keeps a variable, like temperature, very close to where it is supposed to be. Because if it goes too high, there are ways to bring it down. And if it goes too low, there are ways to bring it up. So feedback mechanisms alter the original changes that triggered them. They are called feedback loops. So a great example of homeostasis in the body is temperature. If you are too warm, homeostatic mechanisms will cause skin blood vessels to dilate and sweating will begin, and that causes the loss of heat, bringing temperature down. If you get too cold, your skin blood vessels will constrict and you will begin to shiver, which releases heat energy. So you generate heat and bring your temperature back up. And this is a simplistic way to look at it, but it's very much like a thermostat in your house. And you set your temperature at 65. If it goes up a little bit, your AC will kick in. If it goes down a little bit, your furnace will kick in. Blood pressure, when you rise from the bed, blood drains from the head, blood pressure falls in that region. It's detected by receptors known as baroreceptors. They send information to the cardiac center of the brain, and the cardiac center sends an increasing heart rate to raise your blood pressure and restore your homeostasis. That baroreflex illustrates the common components of a feedback loop. There's a receptor that senses change, that would be the baroreceptor. There's an integrating or control center that processes that sensory information and makes a decision, directs the response. In this case would be the central nervous system, the cardiac center of the brainstem.